Okay, so we're going to go ahead and get started with our second panel for the afternoon. This panel is entitled Data Empowering Social Justice, and we have three very qualified people to speak in this area, so really looking forward to their presentations. First up is going to be Dr. Teresa Cully, and her laboratory is working to better understand if scientists in developing countries where the majority of plant biodiversity occurs are receiving proper got a phone call, sorry, are receiving proper scientific recognition for their research in the form of authorship and peer-reviewed literature. They are also interested in promoting shared, accessible data that will be used in future studies for novel advancements in the biological field. Her research thus further indicates that many scientists in developing countries are not being included in the published literature as authors. So please welcome Teresa Cully.
All right, thank you, Amy. I have to warn you, I'm a wanderer, so I'm going to try to restrain myself to this podium so you can hear me. So thank you very much for the invitation to talk with you here today. And what I'll be talking about is not just work that I alone have done, but actually it's been a team effort. And so I first want to thank Amy, who's the one who eventually got us all together several years ago. The rest of the individuals up there on the top row, and then also Maria and Rob on the bottom. This is a group of students and faculty in Department of Biological Sciences, and it sort of started out with a seed of an idea, and it sort of grew, and more people wanted to get involved. And then also we work very closely with the Digital Scholarship Center. So the next group of people you probably know about or you've seen around. And so the research that I'll be talking about here today is one of the main projects funded by the Mellon Foundation through the Digital Scholarship Center. So we are entirely grateful to the Mellon Foundation for their support. So let's go ahead and get started. Well, if you think about publishing, as a researcher is where I'm coming from. I'm a plant biologist, and I'm also an editor of a journal, and that will come into play in a little bit. But when we think about the steps of publishing, most of us think, oh, you know, it's pretty basic. You have things where, as a scientist, you have to first identify your research question, then you have to actually do the study, conduct the research project itself. Then finally, once you have that done, you need to write up your manuscript. And finally, you need to go ahead and submit that to a peer-reviewed journal. Hopefully you have some idea already of a journal it might be. You go ahead and submit that. You wait with eager anticipation. You're going to wait for that review feedback, and hopefully when it comes back, it's good news, right? So they might accept it. You might have to revise it a little bit, or maybe you have to resubmit it to another journal. But you go ahead and you try. You just keep working through it. Eventually, your manuscript is published, which is great. So this is sort of what here in our society we get used to, and this is as an academic, this is what I train my own students to do. I have many of them who publish with me as co-authors, and then hopefully one day after they graduate, they'll go off and publish on their own. However, once you start thinking about this, this perspective is not the same for everyone all over the world. In fact, I would argue, and you'll hear me argue, that authors, especially in other countries, do not go through this whole process, or rather, they encounter difficulties during this process. So I wanted to talk about some of those. Now, the reason I got started and interested in this, I mentioned before, I'm an editor of a journal, and I've been an editor for a journal, Applications in Plant Sciences, for a number of years, and a co-founder of the journal. And I started to keep an eye out for who was submitting manuscripts to our journal. We also have a sister journal known as the American Journal of Botany. It's been here for at least 100 years. We just had our 101st birthday, so it's the sister journal. My journal has been around since 2013, so we're a little newer. But when I looked at some data, and this might be hard for those of you in the back, but the point is, if we look at the number of articles that are published in 2017, I just grabbed that, and I looked at by country. And here I'm looking at the country of the corresponding author. And if you look, the majority of them are actually on the left are from the United States. And as you go down, there's other countries that pop up there. So we have Canada, we have Spain, we have Brazil. Towards the end here, we have India, Chile, and some other countries, Argentina, for example. Now, and so I was interested in these other countries. Why aren't those numbers higher? Now, we can also go and look at my own journal, Applications in Plant Sciences. This is an online-only journal. Started as just an online section of American Journal of Botany. It was so successful, we spun it off into its own. And here, it's sort of the same old story. We have the majority of publications here from the United States. Next up is China, Japan, Brazil, Germany, the UK, and so forth, and it goes down. So again, I was interested in, okay, there's these other countries, and they're not as represented as much as I feel that they should be, considering that these are, we consider these to be international journals. So that, to me, that there was something happening there. Here's another data set I just pulled yesterday, and this is from 2018. This is Applications in Plant Sciences, the same thing, but here, what I've done is I've looked at the total number of submissions that we've received for the whole past year. We received them from 29 different countries. They're all shown here on the x-axis. But what we have also is within each, I have whether that submission eventually was accepted as a published article or whether it was rejected. And so one interesting thing is that for 2018, China actually contributed the most papers. But if you look at United States was second, but we have an equivalent number of Chinese and North American, and that is the United States papers, accepted. So that was interesting. And then we have all these other little countries that start popping up here. We have Finland, Mexico, Turkey, Thailand, Algeria. And for some of these different countries, we have gone ahead, you'll see a green, that's one paper there that we've accepted. Other countries, Bangladesh, we had one paper that we rejected, and that could be rejected for whatever reason. So looking at this, I started to get comments too from reviewers where they would review a paper from another country and they would say, well, you know, it's not that good because it's a non-native speaker. I'm like, that's not really a good reason. So that was sort of bothering me. And at the time, I was talking with one of my graduate students, and she went to an international conference. She works with the Cincinnati Zoo, and she went to a conference on plant propagation. She came back, and she said, you know, I had this really cool conversation with some scientists. I think they were from Trinidad, and they were complaining that they're never able to get their papers, their articles published in Western or English-speaking journals because of this perception that either they can't speak the language or maybe their research techniques are not as great as what they should be. And so because of these things, a bunch of our lab members got together and we decided that we wanted to look it over. Oh, the other thing I want to mention, that in several other countries, like China, for example, the salary that an individual makes is actually tied to the number of publications they have in English-speaking or English-printing journals. So that is important to think about. Okay, well, the other really important thing, as a biologist, I started to think, okay, we got this problem, and then I started to think about where these authors are coming from that I'm hearing about these problems. And many of them are from tropical locations. Now, in terms of biology, this is really important because it's in those tropical locations that we have the highest biodiversity. So shown here is just an example of birds. I had to put a nice toucan there because he's cute. But what we have, this is a type of heat map, if you will. So if colors are red or yellow, that means it's the highest levels of biodiversity. If they're blue or purple, it's the lowest levels. So what you can see right here, here's the equator. And so you see a large number of the brighter colors are around the equatorial region. And then, so that's birds. Then we can go and we can look at mammals. Guess what? Pretty much the same thing. You see a high level here of color. And then we can go to reptiles, pretty much the same thing. Now, I was trying to get to plants. We have to go to a different data set, so it's going to look a little different, just to warn you. But we can go look at plant diversity. In this case, well, we're talking about actually species richness, so number of species. And here's plants. But really, the thing is the same. If you look at this map, we actually have the highest number of plant species, really, if you focus on South America, in the equatorial region, and that's exactly the area where these researchers are coming from that are trying to publish in journals such as the one that I oversee. So this is a problem. Here's just another example. We can compare United States and Brazil. They're actually very similar in overall land area. United States is a little bit larger. In terms of species richness, R, you can see the United States has 462 species. This is just an estimate, by the way. It's probably off. Brazil has more, 521. However, if we go and we look at little Ecuador, which has a much smaller land area, but yet has 1,856 species, and these are plant species. So something's going on here. So what we started to think about is if we have areas of the world that has the highest biodiversity, but yet these usually have the lowest economic resources. So it's creating this problem that we're seeing. Here's just an example of what I'm talking about with lowest economic resources. This is some data from, let's see, the International Monetary Foundation, IMF. And here we have the global gross domestic product per capita. And this is just based in 1980. The point is, as things get more blue, it means that people there make they have a higher income per person. And so here, this is in 1980. You can see where it starts. We go to 1990, the United States just started turning blue. We go to 2000, the United States got bluer. Here's 2010, oh, Russia's starting to pick up. 2019 today. So one thing you might have noticed is part of South America, which is where we've been paying attention, has started to become bluer, in other words, but not quite up to the level of the United States and Europe, and Northern Europe, and also Australia. Okay, so what we decided to do was, in order to look at this, we had to separate out the countries. So we decided to call them developed and developing. We don't like that terminology, but it's the best that we can come up with. So in order to figure this out, we had 213 countries. Originally, we used criteria established by the CIA, yes, the CIA. We ultimately decided we didn't like that so much, and so we created a new category based on data from the United Nations, the World Bank, and the International Monetary Fund. And so essentially, we have 87 developing countries shown here in dark green, and 126 developing. So we have, again, 87 developed, I got that wrong, developed countries, and 126 developing in yellow. And I also, in some of these countries such as China, India, and Brazil, I colored a lighter green because they are really technically still developing, but they are really rising up fast to the level of a developed country. So, in terms of challenges that these scientists from these developing countries might have, you can think of some of them off the top of your head, and we already talked about some already. But one, obviously, is a lack of fluency in English, or the perception of that. I've had friends who are English speakers, but yet they get a job in China, and they found some manuscripts rejected, and it's written on them, you know, not a native English speaker, and they are. So that gives some idea of the prejudices and the biases out there. Many of these people, as I mentioned, the researchers I know in these other countries have reported having problems getting their work published. And then also, one of the important things for us from the scientific side is that because of the low economic or the limited economic resources in some of these places, it's just really difficult to have access to these modern genetic methods that a lot of magazines and journals, they really want to see its modern work in order to publish it, right? It's at the forefront of technology and methods. And so an example here is if you just take any type of new method. So in my world, next generation sequencing is now starting to get old, but it's still new, and what happens is it's costly. So anytime a new method comes out, initially, it has a really high cost, and then over time, it gets cheaper and cheaper and cheaper. And so eventually over here, this is where the researchers from these other countries, these developing countries, are able to finally use it because it's cheap, it's inexpensive, but yet, if they try to publish that method, then editors might say, that's old. You need to use something newer, i.e. more expensive. So they're sort of stuck in this conundrum. So what I was interested in is, has this inequality in resources, as well as, you can throw in there the language differences too, has this led to a publication bias? And one of the, I think, a really great example is, this is the USDA plant explorer, Frank Mayer. And he was born in 1875. He worked for the USDA pretty much for his whole life. He eventually died in 1918 in China. And he, by the way, is the reason why you have apricots and most of your fruit trees here in the United States. He actually brought them over from China. And we have extensive travel records. He has really beautiful photographs that he sent. Here's some of his photographs. And these are all available at the United States Department of Ag library. And these are all accessible online. It's a beautiful resource. But one thing you'll notice is he has this guide in many of his photographs. In fact, I would argue that Frank would never have been able to do any of the amazing work he did without having these local guides, is what he would call them. But this is someone who would take him to these places, show him where these plants were growing, talk with the local people to enable him to actually come, because here is, you know, this man who obviously did not belong in this area. And so these guides were so important. But yet, we have no idea who this is. I can probably dig up his name in one of the letters here. But he's not really known to science. Okay, contrast that with Harvard. And so at the Arnold Arboretum at Harvard, Professor John Jack was really important and influential in the early 1900s. And so what he did was something different. So Professor Jack actually went to China, identified promising students, brought them back to Harvard, and actually educated them there. And so we know today many of these scientists, eventually they were trained here, they went back to China and to the universities there, and they trained a whole new generation of scientists. And this cooperation between China and specifically Harvard continued today. And so this is one of the reasons, for example, why we have the Dawn redwood. That's a really beautiful tree that we can plant around here. It's related to the giant redwoods in California, but it's originally from China, so we can thank the Chinese for this. So really, do researchers in developing countries receive proper credit for their work? Here I'm talking about authorship. And so we can ask, why should you be an author? And here's some criteria from my journal. And someone is an author if they substantially contribute to the design of an experiment, to the data analysis of the experiment. They need to be involved in drafting the manuscript or contributing it in some way, or at the very least, approving of it, right? Oftentimes you have to sign off on that copyright form. And finally, I would also say that an author is someone who's participated enough where they're able to take public responsibility and really talk about what they did. Maybe they can't talk about the whole experiment, but they can say, I did this piece, and this is how it works, and this is how it relates to the main study. But the best, I think, case for being an author is this one. The project could not have been done otherwise without that specific person's contribution. If you could say, okay, I could not have done it otherwise without that person, then they really need to be an author. So that's sort of like my gut way that I define authorship. Okay, well that's all well and good, but how about non-researchers? Up to this point, I've really been talking about researchers, like academics, like having a degree behind your name. Well, what about non-researchers? And this came up recently. I've been working with a collaborator in Colombia, and she mentioned that at least in Colombia, there's actually a debate on how to recognize these individuals. She calls them para-scientists. And so what they're defined of is members of an indigenous, it could be an indigenous tribe or local communities. They have substantial knowledge that's really necessary, but yet they don't have formal academic training. So this is Dr. Nicola Flanagan, who actually will be coming here to UC in October. And so this is an example where she really believes firmly that we need to acknowledge the contribution of these parascientists. So she discovered a new species of vanilla. This is the orchid. So if you know vanilla, it comes from an orchid. There, you guys have learned something important about plants. And so she actually included people who came with her out in the field to show her the plants. And so this is another example of why authorship can be so important. Okay, so what did we do? So we had all these questions. And so my group, we decided we first wanted to look at the countries. We divided them into the 87 developed and 126 developing countries. And then what we decided to do is a big question. So we decided we're going to focus on plant genetics, and we're going to look at conservation genetics of plants. And so what we did was for each of these countries, we made a big data set. So we had, for each country, we had the land area, that's easy to get, right? The gross domestic product, or some estimate of monetary value, that's pretty easy to get. Species richness, number of species, that's sort of easy to get. And then diversity, and you'll see that's a little bit harder. Diversity, okay, so number of species is you just count number of species there. Diversity is a little different. What you're doing here with diversity is you could have, in this case, we have the same 4 species present at 2 sites. So the number of species is 4 in each case. However, the abundance of these really vary. So in the case over here in community 2, you can see that it says 80% of the species are like this really weird, what the heck is that, an S tree? And we have a few pines and maybe a fir and something else weird, maybe a cedar. Over here in this community, they are all equivalent numbers, 25% of each of the 4 species. So we would say that on the left, that group is more diverse than this group over here on the right, where it's really mainly one species that's spreading all over. Okay, so that's what we're talking about diversity. And initially we thought, oh, it'd be so easy to measure diversity. We'll just go to some data set and we'll find it. Well, guess what? It doesn't work that way. It's not as easy as it seems. And so we began with several different measures, because first of all, it's really hard to find diversity by country. Some countries will have that figured out, others won't. So we ended up using several different methods, and this is ongoing. We're still trying to figure out which is best. But we first used the GBIF. This is the Global Biodiversity Information Facility. We thought at first that that was the right way, and then we started digging down, and we're like, that's just species richness. So we're now using a measure that the United Nations has calculated for diversity. And then Rob Tunison, one of my students, actually came up for his master's thesis, another method, and this is this average taxonomic difference, AVTD, and so he's developed that. And then we can divide species richness by area, and another one is you could calculate the number of threatened species that you can get from the United Nations, divide it by the GBIF richness. So there's all these different measures. So again, the point is that it's not as easy as we thought to actually just calculate diversity. So finally, what we've done is we have publication data where we have plant species, name and country where it's native. And so again, we're looking through the data now. And we can find the country of the first author, the country of the corresponding author, which is usually the first author, the country of the last author, and of all other authors. We've been using Scopus. It's free, which is one reason. It is limited. Data is from 2004 to the present. But with this, we're able to get the abstract and all associated metadata. And you have millions of titles. So that's really nice. We did want to get Web of Science, but truthfully, it's a little expensive. It has been purchased by Clarivet Analytics. It used to be Thomson Reuters. You guys probably already know that. And the range is much greater, 1900 onward. And they have a lot more abstracts and metadata. However, it is a lot more money. And so that's one thing where we're limited at the moment. But that is where we would like to go. So here's just a brief example of what we did. So here's a paper. This is on chamoya. So if you guys have seen this, I've seen it for sale at Meijer. It's a fruit you can buy, and it's native to Ecuador, Peru, and Bolivia. So here's a paper, and we have all these authors and places where they're from. So we want to say, okay, are any of the authors from the location where the plant is from? And the answer is yes, actually, sort of cool. It turns out that we have authors here from Ecuador, Peru, Bolivia, actually Colombia is there as well, and then also Spain. Let's see, we had Ecuador already, Peru, and, Belgium, that's who and Kenya. Kenya, interesting. So this is showing there's this global network. Okay, that's good. Here's another example. This is Cacoa, Theobroma cacoa. Here's another thing you can learn about plants. Chocolate comes from a plant. This is it. So here we have, it's actually a tree. Here's these pods. You could pull these out and process them for chocolate. So here's one where chocolate is thought to be native to Honduras and Nicaragua. However, I was informed this morning by my collaborator, Eric Tepe, that that is now in doubt, and it's probably Western Brazil. This is taxonomics, or systematists like to argue about these things. So if we look at, okay, where do the authors come from here? So they're actually going to come from the United States, China, and what is that, France? France. So these are the results we have thus far. So far, we've been able to look at authors from where they're coming from. And we can see, this is hard to see, but most authors are coming from United States, Europe, China, and Australia. And then if you look at where the species come from, they're from all over. Usually, you can see there's a whole number of them in Africa, South America, a few, some in North America, that's true. But we're seeing this mismatch of species from North America, or from the northern developed countries, and where the species typically occur all over the world, but especially in developing countries. So one of the last things I just want to mention is we're also doing a semantic analysis using JSTOR data now. So we're actually able to download all the articles and search through them for certain word terms. And so that's something we're doing. And we're doing this by country. And finally, I just want to end with what we've learned so far. Obviously, collaboration is essential. Access to data sets can be really challenging, as you've seen. And in some cases, some statistics, some indices are just really difficult to come up with. But finally, really, the message that we have so far, and again, this is an ongoing project, is that there is a publication bias problem. We feel pretty strongly about that worldwide, but we still don't yet know the full extent. But there is something there. So I'm going to stop at that point and see if we might have any time for questions or so. Thank you.
Thank you. I think we'll have time for about 2 or 3 questions.
Hey, Professor.
Yeah.
My name's Frank Walmsley. I'm with the Children's Hospital,
Hi, Frank.
Cincy, and I followed the publication disparity between like the plant species, where they're being found, and then who's actually publishing articles on those plant species, being a disparity. But I don't know if I see the publication bias other than, the non-native speaking publications may not be as highly rated in the review process. So maybe can you speak a little bit more about that? Maybe it just went over my head.
Yeah, no, you're hitting on a really big point. And I sort of skipped over a bunch of things. But yeah, what I was showing you today is that a lot of in terms of what's being published, is not necessarily coming from those countries that we would expect it to. In other words, the authors don't follow, if you have a plant being published on in a developing country, we don't have the authors we would expect to be. Now, in some cases we do. What you're probably getting at, and I think is a really great thing, is why. And so, and I sort of jumped the gun a little bit. I said it's a bias, right? And that's more of a directional type of thing. And that's really where we're at right now, is why. And I think a large part of that is the English component. The other part is a lot of these authors tend to, if they can't publish in an English journal, then they actually publish in a journal in their own language, and that's not even captured here. So you are right that the next thing is why, which is really the bias part. Is there actually a definite bias against that? And I'm going with there is a bias primarily because of the anecdotal evidence, the stories that we've been told. But you're totally right that in order to definitely say there's a bias, we need to show that. And that's where it's very difficult. So I've been working with Wiley, my journal is associated with Wiley, to actually get the inside data. And we've also done, last thing I'll show you, we did a survey of our members. And one of the things we're asking, which influenced your decision on where to submit your journal. And this one is interesting. It says science is done in premier institutions, but also under difficult circumstances in far corners of the world. But then later they say, I judge this by knowing whether most of their papers come from only developed countries. So to me that has a little bias in it, and it appears to be from an author from a developing country, which is weird. So part of what we're doing is we're publishing on the science, and then we're also working on opinion pieces, sort of pushing the academic world to start thinking about these things, in other words, to push it a little bit more forward. I'm not really answering your question, but that's essentially what we're trying to do. So I'm probably out of time, looking at Amy.
Other questions? Maybe we can catch Dr. Cully afterwards. How does that sound? Okay. Thank you. So our next speaker is Brian Howe from UC College of Law and the Ohio Innocence Project. He's going to be talking on the topic of data and wrongful convictions. The Ohio Innocence Project is a legal clinic that seeks to overturn wrongful convictions and free innocent men and women from Ohio prisons. The Ohio Innocence Project also seeks to educate the public about the causes of wrongful convictions and enact policies to reform the criminal legal system in Ohio. To date, the Ohio Innocence Project has helped free 28 innocent people from prison, making it one of the most successful post-conviction clinics in the country. So, Brian, look forward to your talk.
Thanks, Amy. Thank you, everyone. I'm grateful for the opportunity to be here and grateful to talk to you all about data and wrongful convictions. I'm an attorney, not a researcher. I won't be able to point to much data research that we've been able to do at the Innocence Project. We're mostly focused, again, on actual casework. Before I get into it, I really do need to speak and do a little bit of a plug for the Innocence Project. This is a legal clinic. We have 16 to 22 law students who are working on real cases of people in Ohio prisons who are claiming wrongful convictions. We've reviewed so far in the last 15 years about 10,000 requests for assistance. These are reviewed by students. Students are doing a lot of the actual legwork in identifying wrongful convictions. We then take those cases that we can litigate and move those forward. To date, again, we've exonerated 28 people from Ohio prisons. Represent over 400 years worth of wrongful incarceration among them, making Ohio one of the most successful clinics in the country like this. I think we're third right now, maybe behind California and Chicago, so it's something that I'm really proud to be a part of, and I think that we can be proud of at UC. These are our clients, Derek and Larice, who were released just a year or two ago, and recently had a victory in their civil suit. So, again, we are litigators at the clinic, and so our intersection with data is really how this affects what we do, and it happens in mostly two ways. And first of all is the scope of the problem. How big is the problem? How many people are actually innocent who are in prison today? And the second is what causes the wrongful convictions that we know about? What is the source of these mistakes in the legal system, and how can we work to correct those? Dealing with the first question is extremely difficult. I think you can probably imagine right away you're going to run into some really thorny, deep epistemological questions, right? The problem is, of course, that determining who's innocent and guilty is what we expect a trial to do in the first place. And it becomes really hard to get to a sort of ground truth behind that. The only way that we know that we make a mistake is when we're able to have some success through the same legal system that was responsible for the conviction in the first place. So we sort of know about the mistakes that the system allows to be brought to light, and flaws in that system can actually make it harder to find the flaws in the system in the first place. The denominator is pretty easy here in this equation. So, right, if you're trying to find the rate of wrongful convictions, it's the number of people who are wrongfully convicted over the entire number of convictions in the United States. Lawyers are not great at keeping track of data, but we do have at least some data about the total number of people in prison. On a yearly basis, it works out to be about 1 million people put in prison every year on felony convictions, which is a tragedy in and of itself. But finding the numerator is a lot more difficult. We know that people are wrongfully convicted. As of this morning, I think it was right around 2,500. There's a national registration of exonerations that tracks these things. It's something close to that, and we can assume probably that that's not the entire universe of wrongful convictions. We haven't solved every single one. I still hopefully will have a job tomorrow and things left to do and convictions left to be challenged. But how do we extrapolate from those 2,500 to the entire universe of convictions? It requires answering some really, really difficult questions. First of all, there are some types of charges that are more likely to lead to wrongful convictions in the first place. Second of all, there are certain types of wrongful convictions that are more likely to or less likely to result in exonerations. So there are certain types of convictions that are particularly difficult to challenge in the system. And third, how do particular laws in our respective jurisdictions. We have most criminal laws done on a state level. We have 50 different states, 50 different laws, 50 different post-conviction laws, 50 different DNA testing laws. How does that all impact? And so right away we get into really thorny and difficult questions. There are some less than ideal ways to answer this, and sometimes people just sort of use their gut intuition, including, this is the late Justice Antonin Scalia, who had famously sort of cited this is not actually his words, he cited this in a capital case, sort of dismissing the idea that this is really a big problem to be dealt with. At the time, he's referring to a 2003 study that at the time identified 340 wrongful convictions. It was actually still fairly early on in the Innocence movement, and said, well, let's just be really, let's be crazy and assume that maybe we're only catching 1 out of every 10 people who are actually wrongfully convicted. And we're only reversing 1 out of 10 of those. We're only identifying 1 out of 10 of those. Well, even still, then, you have 15 million convictions across those same time. And so here we go, a 0.027% error rates, 3 significant digits for his about 10% assumption. And so a success rate of 99.973%. And he uses this to sort of suggest that this is not a significant challenge or problem in the legal system. And so there are some problems with that. And I think one of the biggest problems, there's a lot of problems with that actually, but the one that I want to focus on is just this sort of assumption that maybe we're only catching, we're only catching 1 out of every 10 wrongful convictions. And 1 out of every 10 wrongful convictions is being corrected, identified by the legal system and corrected. You might think that that's a depressing number. Justice Scalia thought that should be sort of a on the far end, low end estimate of what we're catching and correcting. I think it's probably high. I don't know that I would pop champagne corks if it turned out that it were actually 1 in 10, but I think it would be actually cause for me being more optimistic than what I am now. And the reason is that a lot of things, these are very low probability events, exonerations are. And it's because you have to get really, really lucky to be exonerated once you have been wrongfully convicted. Evidence still has to exist. You have to be in a place where there are procedures that will allow you to identify and discover that new evidence, and you have to be able to raise that new evidence in a context where the mistake is then going to be corrected. At every stage of that, it gets less and less likely. And just in case if you think that I'm making up how difficult it is, this is a quote from the Ohio Supreme Court about the post-conviction process. And again, this is actually sort of a response to mass incarceration. As we continue to incarcerate more people, the legal system has made it more and more difficult to challenge these convictions post-conviction, really for the purposes of reducing their own caseload. And this is the Ohio Supreme Court sort of saying the quiet parts out loud, that some people have said that the way that the laws governing post-conviction review are basically a virtually futile review process. To the extent that we have a lot of frivolous claims to deal with, sure, we're going to not necessarily deny those assertions. So this is a very difficult question to deal with. There are people who have made, I think, good attempts at it. The first, or I think probably the most compelling in my mind to date, is a study by Gross in 2014. This looks at death row cases. It examined 7,482 death row cases between 1973 and 2004. The reason that it focused on death row cases is that the idea is that these are cases that receive a particularly high degree of scrutiny. Generally, you have death row defendants who are on death row will have legal representation all the way up until an execution. And so if anyone is going to have success in challenging a conviction, it should be someone who's been convicted of a capital crime and sentenced to death. It actually used a fairly sophisticated survivability analysis to take into account people who are removed off of death row for reasons unrelated to exoneration, so people who die of natural causes, people whose sentences are commuted and therefore would reduce some of the pressure and some of the resources available to them to challenge their convictions. That's all taken into account by this study. And what they ended up finding was they're assuming about 4.1% would be exonerated if you had taken all of those people and left them on death row up until the end of the sentence. So that's probably the most sophisticated attempt to measure this. There's another study that's more recent out of Pennsylvania that talks about self-reported wrongful convictions. It analyzed 2,600 people entering Pennsylvania prisons between 2015 and 2016. Gave them an anonymous survey where they were to report back whether they felt they were wrongfully convicted or not. And it wasn't just, are you innocent or are you guilty? It's not like Shawshank Redemption, actually. There's a very small number of people in prison who are asserting their actual innocence. And among people who are challenging their convictions, it gave them a pretty wide range of choices. I didn't do any of the things that they convicted me of. I did some. I did it, but I didn't get a fair trial. I was involved, but maybe it was justified. The only thing that they used for the numerator there is that very top answer. I didn't do any of the things at all that they convicted me of. And then they adjusted that downward a little bit to result for some cases they thought were maybe less than credible. And the answer that they came up with as sort of a top line, it's probably unlikely to be overestimated, is between 6 and 8% of people in that study. You had a very high response rate, by the way, too, 94%. And they broke it down by crime as well. You can see sexual assault there on the very right-hand side seems to be a bit of an outlier. But for most of the others, it's right around that 6% mark, give or take. People are claiming actual wrongful convictions. So the bottom line here, unfortunately, is that we really don't have much of an idea. It's probably somewhere between 3 to 5%. There's a very high degree of uncertainty here. But it's important to think about, again, we're talking about 1 million felony convictions every year. If you have 4% of those wrongful convictions, that's 40,000 people every single year. That's an epidemic proportions. There are 20,000 homicides in the entire US every year. This is twice that many potentially being innocent people being convicted of crimes they didn't commit. The other thing that we can talk about with maybe slightly more certainty is some of the factors that lead to wrongful convictions. And this starts to get into how we at OIP are able to start using this data. We can use it regardless of really how you extrapolate the known instances of wrongful convictions to the prison population generally, because sort of intuitively it makes sense. If we have several thousand examples of convictions going wrong in certain consistent ways, we can start to assume that maybe there's something methodologically wrong with what's happening in these cases, and in fact that is what we find out. All of these tend to be pretty reliable indicators of systemic flaws or cognitive biases. For those who can't read, on the very left there it is, or can't see the small print here, it's mistaken witness ID. The second category is perjury or false accusation. In the middle is false confessions. The green bar is false or misleading forensic evidence. And on the right, it's official misconduct. And this is, again, covering the 2,500 exonerations known to date. Since it's Data Day, I just wanted to say a few things about the false or misleading forensic evidence. So some of you may have heard a few years ago, the FBI released a report showing that of the 28 analysts that it had reviewed. This is the FBI, so Quantico has a lab. They send around their hair examination experts in cases where they're really supposedly the sort of big guns being brought into these cases. They're supposedly the best in the country. And as they went and audited these cases, they found out that almost all of them overstated what the conclusions should have actually been in favor of the prosecutors 95% of the time. 95%. They were wrong in favor of prosecutors, or it was testimony that wouldn't be supported today. Of those 268 trials, 32 of those were death row cases. 14 of them have already been executed by the time that the FBI released their study. So that's not something that's unique to microscopic hair comparison. We have, in 2009, the National Academy of Sciences issued a report, and I'll quote from that. It says, much forensic evidence, including, for example, bite marks and firearm and tool mark identifications, is introduced in criminal trials without any meaningful scientific validation, determination of error rates, or reliability testing to explain the limits of the discipline. That was actually confirmed, was controversial at the time. It's since been confirmed in 2014. The Presidential Council Advisors on Science and Technology quoted that and sort of cited the, quote, historical reality that many forensic methods were devised as a rough heuristics to aid criminal investigations and were not grounded in the validation practices of scientific research. This is sort of a problem that's being more and more recognized in criminal prosecutions. A lot of the forensic sciences haven't had a lot of scientific validity, aren't necessarily as valid as maybe they should be for issues that are really, really important. People's lives are on the line here. The other thing that I'll say about the causes of conviction is, again, although the fact that any of them are on this list is enough to probably indicate that it deserves some attention, when we start to look at the proportion of each of these factors in wrongful convictions, we run into a lot of the same problems, epistemological problems of knowing what we don't know, really, or getting to a ground truth behind wrongful convictions. And you can see this somewhat in the differences between how these factors are seen in types of crime. Sexual assault cases were originally sort of the main source of data that we had. The entire Innocence movement was really grew up around DNA exonerations and the ubiquity of DNA testing, the ease of DNA testing in cases, and those happen to be cases that are, sexual assault cases are very prone to audit by DNA testing and forensic analysis. So you have those on the right. Homicide cases are not always, you won't always have forensic evidence that you can test after the fact. You won't always have that kind of ability to scientifically go through after the fact and check the conviction. And what you see is that official misconduct on the right-hand side really is the either equal to or second after perjury or false accusations, which, by the way, are mostly incentivized testimony, prison snitch testimony, people who are testifying in order to get better deals for themselves. I don't know why official misconduct is necessarily so much higher in homicide cases, but I think one of the reasons may be that a reflection of the procedures that people can use to get exonerations post-conviction. A lot of people don't know there's not actually a constitutional right to, for example, a new trial in the case of newly discovered evidence. What we have constitutionally are procedural guarantees. What we're guaranteed is a fair trial. We're guaranteed as part of that certain things that go along with our conception of a fair trial. But assuming that you have a fair process, that's usually where the law leaves it. They don't want to relitigate cases endlessly. We don't want to have second trials or third trials. As long as the process was fair, that's what you get. And this is, again, Justice Scalia, I think, maybe trolling a little bit. This court has never held that the Constitution forbids the execution of a convicted defendant who has had a full and fair trial but is later able to convince a habeas court that he is actually innocent. So in other words, you can convince a federal court, district court, that you are actually innocent of the crime that you were convicted of, and in Justice Scalia's opinion, nothing about that necessarily prohibits your execution. So the official misconduct cases are the lever, I think, that a lot of people use to challenge convictions to come back through, and that's maybe why we see those overrepresented among homicide cases. So we're generally using this data on sort of the causes of wrongful conviction to identify cases to work on and to litigate these individual cases. We're also using them to try to reduce the incidence of wrongful convictions moving forward. One of the things that you all would probably be surprised about is how methodologically sloppy criminal investigations tend to be. If you all do research here, you probably have very basic protocols about being blinded to the results, just very basic procedures that are going to limit, hopefully, the sorts of confirmation bias that all of us are prone to. That doesn't really happen in a criminal investigation. And so one of the things that we've done is worked with the state legislature to pass a law requiring some changes in procedures for photo lineups. This is one of the places that there tends to be a lot of errors in witness identification. And so what the 2010 law did was, instead of viewing this is sort of the traditional way to conduct a lineup, it's called a six-pack. You have your suspect somewhere, one of these six. You show the person the six-pack. It's usually the detective who's actually investigating the case, and the subject, then witness, picks one of those six. The problem is that when you have the person conducting the experiment doing the lineup, they're prone to all sorts of subtle ways of indicating things towards witnesses, and we find that it's really ends up being a fairly biased procedure. The law that was passed in 2010 requires law enforcement to present pictures to witnesses one at a time, so you're presenting them sequentially instead of all together. It's done by a person who has no connection to the case, so it's a blind administration of the lineup. You're informing the witness that the person is not necessarily in the list of pictures, and they actually have a row of sort of blank pages at the end, so the witness doesn't know that you're getting close to the end of the list, which can actually encourage them to just pick someone if they think they're running out of options. Those very simple procedural things can really increase the accuracy of identification. One of the things that we're also doing, and this is sort of still in progress, is trying to gather data and apply it ourselves in Ohio, specifically around forensic testimony. The reason this matters is that you have, as we talked about, forensic testimony changing a lot over the last few decades, and that's a process that's still going on right now. The fields are still evolving, the conclusions that they believe they can draw are still evolving. And when it comes to raising this in an individual case, the problem that you run into is that the law requires you to categorize this as newly discovered evidence. And what the law is contemplating when it's talking about that is sort of a specific concrete piece of evidence that you discover and then you raise within a reasonable amount of time. And of course, that's not really, there's no way to really apply that to a gradually changing scientific consensus. So for example, I just made all of these numbers up and conclusions up, but you have a conviction, say, in 1996 on the basis of Bitemark testimony. You may have the first study that sort of maybe suggests some problems happen a few years later. There's maybe responses to that as they circle the wagons within the field and produce studies that purport to validate the methodology. You may have a single court exclude this testimony a few years after that, and then other courts include it after that. And it may be a 15, 20-year process of changing scientific consensus. And where that leaves the defendant who was convicted in 1996 is that almost no matter where you want to challenge Bitemark testimony along that spectrum, the state is either going to say that the science isn't settled yet, you're jumping the gun, it's still uncertain, or of course the science is settled, it was settled years ago, you're way too late and you've waived this issue. You should have brought this up 5 years ago. And really, no matter where you are on this process, the state can sort of have that response. So one of the things that we're trying to do is track individually, forensic analysts track the way that testimony has changed over time, track the way that labs have changed their testimony over time, track the way that jurisdictions have changed their testimony over time. And I don't know that that necessarily solves this issue of changing science and wrongful convictions, but at least it will help us hopefully get a better handle on that. So we're in the middle of this, so far about 500 cases, which is really just the tip of the iceberg. But we're hopefully going to start to get to the point in the next year or so where we'll be able to draw some conclusions. So unless there are any questions, thank you for your time. And again, I appreciate your allowing me to participate.
So I think we have time for one or two questions.
Okay, my name's Preston Frash. I'm a PhD student with the College of Education. And sometimes I've actually done classes on false convictions because it was in our textbook. But I was just wondering if you could say a little bit more about why there are false confessions. I was just curious about why that happens sometimes. I know sometimes there's pressure to get a better sentence, perhaps.
Yeah, I mean, it can mean a few different things. So in some cases, you'll have false confessions that are really just people flat out lying. And it's the same as incentivized testimony. So you have someone who is a co-defendant. If the police think that a couple of people are involved, they'll find the first person and say, if you cooperate now, you get the full walk, and we're going to go after other people. So in that case, it's the person understands that they're lying, and they're lying specifically because they're afraid of being subject to the most severe penalty. The more interesting sort of false confessions, or I guess maybe the other type here, is the instances where police can pressure and coerce someone to admit something that they've done by themselves where it turns out to be false. And this happens in a lot of subtle ways. The read technique, which is the most common technique, it's what officers have been trained in for decades, is really designed to sort of exert psychological pressure on people over the course of really hours. And the pressure is designed to not necessarily get at the truth of the matter, it's really designed to produce confessions. And it could be something, it could be very subtle in terms of promises. It could be flat out lying to people, saying that we know that you did this. I have a capital case right now involving where a 16-year-old boy was taken to a clinical psychologist, child psychologist, hypnotized, suggested that he had repressed memories of the murder. They took him out to the crime scene, let him see everything, let him see the house. He couldn't identify basic facts about the case, but over the course of several sessions, eventually started to sort of internalize, and he was being told by people in authority that he was responsible for this crime, and eventually sort of made up a story that went along and satisfied law enforcement. So it's a known psychological fact. It definitely does happen, and it's really hard for people to understand or sympathize with, I think, sometimes, but it's something that we know occurs and something that we need to address in the criminal justice system.
Hi. I was just wondering if you guys ever do any work regarding people who have taken plea deals, because I know I've read a lot about people kind of being coerced into taking plea deals because, you know, maybe they have a family at home and, you know, and then they have all that pressure on them, maybe based on income. And I was just, you said that you do other work with like policy. I was wondering if you do anything.
You know, I mean, this is a place where really there could be some reform. Overturning a guilty plea is basically next to impossible. The legal standard is a manifest injustice. Proving that is basically impossible. We almost have to reject guilty pleas at the Innocence Project before doing a lot of research. Unless there's something very clear that we can point to, they're gone. But you're right, that people do plead guilty all the time, and a lot of times it's for, especially in lower level felonies or misdemeanor cases, where if they plead guilty, they know there's a high chance of getting probation, or maybe, you know, some sort of community service sentence. Whereas if they stick to their guns and wait to go to trial, they may be stuck in jail where they can't afford bail for another 30 to 60 days. And really, that punishment ultimately ends up being in place whether or not they're ultimately exonerated. It just makes sense to plead guilty. And that's happened in Houston with drug conviction cases. There's a really famous study, so it's definitely a problem.
Yeah, you guys just don't.
There's not, you know, other than changing the law to allow it to be easier to challenge a guilty plea post-sentencing, there's not really much to do. I mean, one of the easy things to do would be to allow people who had pleaded guilty to access to DNA testing. That's something that Ohio doesn't even do right now. If you pleaded guilty and you want to later get access to the physical evidence in your case to do DNA testing on it, you're completely out of luck. So it's one of the ways that the system makes it harder for people to challenge convictions, specifically with pleaded guilty. And it's all things that we can change legislatively, but there's not right now a big push for that.
Hi. I have a question regarding racial disparities. So we know that there's racial disparities when it comes to mass incarceration in our country. Does the Innocence Project look into racial disparities in Ohio, or have you seen that within your work when it comes to wrongful convictions?
Yeah, that's a great question. You know, on the one hand, trying to sort of get a methodology that would find racial disparities in wrongful convictions just sort of is all of the same problems in finding wrongful convictions, a baseline wrongful conviction number in the first place, and really makes it a lot even more complicated because you've got to sort of address how the sort of racial inequities in the system are affecting these specific cases. And so on the one hand, it's really a next to impossible problem to answer in a real methodological way. On the other hand, you know, it's like the easiest question in the world. Of course, there are racial biases that affect wrongful convictions. We can see it in cross-racial identifications, that witnesses who are a white witness trying to identify a black suspect is much more likely to make a mistake. You can see it in studies that discuss capital sentencing and the fact that white juries are much more likely to recommend a death sentence to where the victim is white and the suspect, the defendant, is black. That same sort of logic should, of course, apply to whether they're more likely to convict in the first place. Methodologically, it's really difficult to address, but you know, we see it every day, certainly, and it's just, I mean, there's no question that problem exists.
Thank you.
So thank you very much, Brian. Okay, our next speaker in this session is Christopher J. Sullivan from the School of Criminal Justice here at the University of Cincinnati, and his talk is entitled Working with Agency Data to Better Understand Racial Disparities, the Case for Disproportionate Minority Contact with the Juvenile Justice System. And this presentation is based on a recently conducted study that sought to better understand patterns of disproportionate minority contact in Ohio's juvenile justice system. The project required extended assessment and integration of record data that varied in their structure, availability to key fields, and operational definitions were collected or extracted from dozens of local juvenile court and police agencies across the state. Currently lead federally funded research studies on juvenile risk and needs assessment and important reforms in the Ohio juvenile justice systems. So thank you, and welcome, Chris.
First, thank you for inviting me, and thanks for everyone to be here. Actually, so what I'm going to talk about today is part of a larger study, as was mentioned, and I'm kind of distilling down a document of several hundred pages to just a few slides. So if anyone's interested in the full report or any of the papers that we've published so far with these data, I'm happy to share. I'd be remiss if I didn't acknowledge the Ohio Department of Youth Services that funded this work, and then also several grad students here at UC who went along for this lengthy ride in gathering up a great deal of disparate data, synthesizing it, and then kind of moving it into a place where we could report some things that we hope are of use to the agencies involved. And so I'll start by just presenting some numbers and background just to give you a sense of the problem, and then I'll kind of go into a little bit of discussion of the data before presenting some key findings across a few different areas. And again, these are just a real small sampling of some of the things that we found along the way, but hopefully they'll be of some interest to you today. So the notion of disproportionate minority contact or racial and ethnic disparities in the juvenile justice system has been an acknowledged issue for some time. If we go back to the 1970s, and then all reauthorizations of the Juvenile Justice Delinquency Prevention Acts at the federal level, they've all contained language on these questions. And this is really driven by the fact that justice agency records typically show racial and ethnic disparities at multiple contact points in the juvenile justice system. However, at the same time, as much research has occurred on this, things are less clear as to the why and exactly what those disparities mean. That kind of goes back to a point that came up just a few minutes ago, or has come up a couple of times, which is the distinction maybe between a disparity statistically and then bias. And I will say kind of at the outset that we don't necessarily have a full answer for that second question either, but what we hope to have done here is to maybe look a little bit more closely and a little more perhaps rigorously at these data in the state of Ohio. And really this comes down to this question of whether or not there's differential behavior that is driving these trends, or there's actually differential treatment happening in the system, and that really does parallel that question of whether or not it's just a disparity or in fact there's some differential treatment happening. One way that we get at that is by looking at all of the possible legally relevant factors that we have access to, and then considering whether or not we still see some of that disparity. And so we'll see a couple of examples of that as we go along in this talk. And so in terms of motivating the discussion, I think one thing just to keep in mind, and it's a good news, in our juvenile justice system, generally, we actually are in a pretty good space right now, in part due to broader behavioral trends in terms of juvenile crime rates, juvenile violence rates. We're seeing historic lows. You can see this trend, this pretty pronounced downward trend from the year 2000 to closer to today. This is good news. This is the byproduct, as I said, of those broader societal trends and kind of what we're seeing. And then also, I would argue that some of it is actually the work of the juvenile justice system in making some changes. In particular, trying to prioritize community-based placements as opposed to state-level residential facility placements, which would be the equivalent of kind of a juvenile incarceration rate. So in some ways, there's positive news, but unfortunately, one of the lurking problems and sort of stubborn problems is that we still do see these disparities. So if we look here in the even ratio line here would be in terms of 1-to-1 as far as non-white and white youth placement in residential facilities would be this black kind of overt line here. But what we see is that the ratios are actually kind of fairly pronounced towards incarcerating more minority youth in the system. And so there's still this overrepresentation that's kind of this stubborn reality. And one of the real manifestations of this problem is that, in turn, youth of color are more likely to experience the negative impacts that we know happen in placement. So even if youth are getting treatment, it's still not necessarily the best environment for a youth to spend several months in. We also know that there's a developmental interruption. In addition to some of other disparities that occur throughout a youth's life course, this is likely to accelerate and sort of make those even more pronounced, especially if we see these disparities. And then lastly, I would say that these youth in minority subgroups are less likely to benefit from the decline that I just showed in terms of not necessarily being able to experience that, and so, or at least as readily. And so there's reasons to look and continue to look at these potential disparities and what they mean in the juvenile justice system. So the purpose of our study was to determine whether there was a problem with disproportionate contact at various points. We started with police agencies and went right through the court decision-making process. We also fortuitously got access to state residential facility data, and I'll show you at least one slide that gives some insight there, which is actually a rare and unique piece of information relative to other studies that have occurred on juvenile disproportionate minority contact. And so the idea was to look at this statewide and locally, and then also to look beyond what could be inferred from that relative rate. So I showed you the ratio of placement between, again, that was a binary coding, and you'll see a lot of binary coding in part due to Ohio's distribution of race and ethnicity, but also in part due to some data limitations. But nevertheless, it was also an idea, or also the objective, to get a little bit beyond that and look at other factors that could condition that relationship. And then where we could, develop some explanations, or at least develop a little bit more insight beyond just the notion that, yes, we see these disparities or do not see the disparities at particular points in this process. So just to give you a bit of an overview, and I won't sort of belabor this too much, this gives you the summary of all of the data that we worked with in the course of the study. It's kind of divided into two pieces. I'm going to focus on the top panel today and show you a few different snippets from there. But ultimately, we worked with 30 or more agencies, 30-plus agencies, to get record data for a minimum of a couple of years earlier this decade, 2010, 2011. This sort of engendered all different forms of data, all manner of data acquisition challenges, conversations on data security, data usage, some situations where agencies were still using paper records and kind of going in and collecting and then coding those and bringing them into our data framework. Dealing in some cases where agencies didn't necessarily have, because of contractual agreements they had made with record management system companies. They didn't necessarily have access to run queries against their own data, so that set up some challenges as well. But ultimately, again, we saw different portions of data from 19 police agencies, 13 juvenile courts, and state facilities as well in the course of the study. And then on the far column there, or the middle column, you can see the sample sizes for each set of records. And then on the far column, you can see some of the key measures and themes that we covered in each of those areas as well. Ultimately, again, as I mentioned, the idea is to get enough information so that we can supplement not just simply looking at a comparison, but also then adjusting. One of the challenges in this case, or in any of these cases, is really to, as I mentioned, account for legally relevant factors. So one of the common responses that you may get from individuals who are working in agencies, and it's not an unfair response, is, show me how this works once I see the different types of offenses that individuals are committing by subgroup. And so we made an effort to try to get as much of that information as we possibly could. I'll show you a couple of different kind of conditional factors that give some illustration that there's still some things that are happening in terms of disparity even after we adjust for those factors. So the idea there is to really to look and compare similarly situated cases as opposed to just compare across the overall distribution, which sometimes we may misestimate in those circumstances. So account for legally relevant factors. We had to integrate multiple sources of evidence. As I mentioned, I won't really go into some of the other pieces of the data today, but we also wanted to observe and hear from people in the system to get a sense of their perceptions. And so we did focus groups with police agencies, and we did interviews in juvenile courts across the 13 counties that we worked with. And then offer some suggestions for policy and practice. Ultimately, though, our objective was to collect and synthesize a lot of this information and answer some key questions. So the first thing I'll show is just some data from the police records, and this is a subgroup or a subsample of the record, or the record count that I showed earlier. But one of the things that we can look at is if we look at these binary categories and we look at weapon involved versus not. So on the left panel here, we can see that there's actually parity at that point. And so, to some extent, that's a situation where if we look just at the surface, there may not be much apparent difference. On the other hand, when we were able to break down to the type of weapon, we actually see a ratio of nearly 2 to 1 for firearms. And there's two pieces that we know in these situations can be important. One is that the reason perhaps to have a firearm, and there's a great deal of research from the adolescent development and juvenile delinquency literature that suggests that there's more gun carrying in dangerous neighborhoods, often for protection. And then secondarily, on the policy side, in the state, we have sort of gun specifications that will automatically up the disposition terms that youth might face as a result. And so this kind of data, this piece of data sort of illustrates that there's a fundamental disparity kind of built in perhaps into sort of day-to-day life that then carries over into the legal structure in which certain youth are kind of coming into the system. And then a second data point just from the police record data is the source of the complaint. And again, this was somewhat limited in terms of the cases that we had it, but one point I wanted to show is that slightly for white youth, there tended to be parent-guardian complaints actually, and these differences are statistically significant, by the way, in a bivariate test, but basically the parent-guardian source of referral was a little bit higher among white youth. But then if we look at school official, the school official panel here, what we see is that there is a few percentage point and statistically significant difference in terms of school official referral. So commonly what we're seeing in the juvenile justice system, and have been for quite some time, especially after Safe Schools Act in the 1990s is the kind of ratcheting up of school-based referrals to the juvenile justice system. And we are seeing some disparities that carry over there, and that being a mechanism into the juvenile justice system. So again, that sort of illustrates to some extent one possible reason for some of the disparities that we see at the surface level. Detention. So we also looked at pre-hearing detention, and really the idea is that detention for youths is an awful lot like the equivalent for jail or no bail status in the adult system. And so youth are held pending their court hearings. That could be based on the nature of the offense. It could be based on the degree to which they may attend hearings or not. It may also, in part, come from the ability of them to have supervision in the community, or at least a perception of whether or not they'd have supervision in the community. And so what we do see is that race had an effect, and we basically control for a number of factors in looking at these differences. And what we see essentially is that in all of these different models, whether detention, and then we go to secure confinement, there is higher likelihood of placement in detention for minority youth as compared to white youth. And what we then see with the bottom effect here that I do want to point out is that basically we often see cascading effects, and this is true in terms of bail status in the adult system as well, where youth who are in detention may face at least a penalty, and sort of some of the mechanisms are a bit complicated, or the suspected mechanisms are complicated. But what we do see is that once youth are detained pending their hearings, that they also have higher odds, significantly higher odds of then being adjudicated delinquent later on, and then also facing secure confinement. So facing that state residential placement that I mentioned early on. And so essentially what we see is that if there's this detention disparity early on, and this is seemingly just kind of a front-end decision that's made at intake, and maybe made and maybe made with limited information, that can have kind of cascading effects downstream that then also affect the degree to which the youth is adjudicated delinquent, and then also the degree to which they are placed residentially at the conclusion of that adjudication. And that does include, all of these analyses include some adjustment for factors that we would expect to influence their adjudication or to influence their placement. The next panel, just quickly, the adjudication piece, this is sort of the overall effect, not the effect that's conditional on detention that I just mentioned. And this one is interesting just from the standpoint of actually the line doesn't come through, but if we look at 1, again, as kind of a central point. So anything above 1 would be considered a worse outcome for non-white youth, and anything below would actually be a good, a potential positive outcome, just from the standpoint of whether or not they're adjudicated. And what we see here is actually that the finding would trend towards a non-white youth having less likelihood of being adjudicated delinquent. And this can be a puzzling finding on the surface, but it's been sort of unearthed in a number of different studies previously. And what we see in our data actually is that this is one there's a lot of variation. And so you can see a couple of counties where, in fact, the trend is in the other direction, and then you see a few counties where it is below 1, and consequently non-white youth are less likely to be adjudicated delinquent. This, in some sense, is illustrative, perhaps, of some of the origins of disparity in terms of discretion, in the sense that adjudication is going to be the place where there's most kind of fact. The facts come mostly into play, more so perhaps than other stages that I mentioned where there's a bit more discretion. So this is a finding that's been identified before, and we're still kind of unpacking it some, but nevertheless it sort of varies somewhat across places too, and really looking county by county as well as at the state level allows us to see a little bit more of that than we would otherwise. This panel really just shows further sort of disparate effects in terms of secure confinement. And so what we see, in fact, is that race had significant relationships with some of the deeper end outcomes that we would see. So state level commitment, I already showed you in some ways an illustration of that. And then also bind over to adult court. So that is a pretty substantial outcome in the sense that that's going to move the youth into the adult system in terms of their, in terms of the possibilities, the possibility of severe punishment for their offenses. And what this is really just showing is the effect of controlling for some of the legally relevant factors, mainly in sort of the really high stake, the highest stakes outcomes. And what we see essentially is that there is a decline in size. So we move from this effect here, 1.8, so about 80% higher odds, and this is for secure confinement. And then essentially this is where we have a full model, so it goes down to 1.12, so it's declined some, but still does not go away fully. And consequently, again, what we're seeing is when we adjust for factors that we really should adjust for, we're still seeing these disparities. And you can see a similar pattern really in the bottom panel as well. And the last piece of data that I'll show you is really from the facilities. And essentially what this shows, and this is a sort of an interesting set of patterns that we reached, or sorry, an interesting set of findings that we reached from the standpoint of, as I mentioned already, a lot of our understanding of this problem stops once the youth is referred to a facility. So once they are essentially placed, a lot of our knowledge, we haven't really gone past that as frequently in prior research. And so what we've done here is essentially looked at a couple of things, a couple of variables together. So we basically have three key data points. We have the groupings that I've talked about quite a bit. And what you can see here is that, you can see it from the N. In terms of our sample, the prevalence is higher for non-white youth. You can also see it in the predominance of the dots in this plot, really. So you see a lot more dots in the left-hand side than on the right-hand side. And then the second thing that we see, if we look at the data, is among the non-white youth, a lot more disciplinary infractions and then a lot more days in seclusion. So in terms of facility experiences, these are both problematic outcomes. We know that there's issues in terms of seclusion time that youth face and adults face who are in solitary confinement while they're incarcerated or while they're in a facility. And a lot of that is driven by the disciplinary infractions that might be accumulated. And here what we have are reports of disciplinary infractions, and they're much higher among minority youth, and then also so our days in seclusion are much higher as well if you compare the left and the right-hand side. And I really just kind of created this window so you could see the number of data points at a fairly high level in each of the respective sides of the plot. And so what you can see is that the right-hand side window is fairly empty, and the left-hand side window is fairly full, even if you account for the difference in the number of youth that are in these facilities. And then the third thing that just wanted to show quickly is the fact that you see this linear relationship. So as the number of disciplinary infractions increases, so too does the number of days in seclusion. And really that's going to be the primary driver in these residential facilities that we have. And so when we see the race disparities in terms of disciplinary infractions, that then in turn affects seclusion time, and consequently, again, minority youth are having disparate experiences in the facilities as well, and that can have further impacts developmentally and kind of on their long-term trajectories as they leave the juvenile justice system and try to transition to sort of an adulthood, toward adulthood even after they're released. So I'm going to leave it there in terms of the findings. Like I said, this was just a sampling of things. There's a number of different smaller papers that we've published. Just recently published a paper. One of the graduate students I mentioned was the lead author on a paper that sort of looks at this particular relationship as a center point. And then we also have some work on, I mentioned weapons offenses earlier. We have some work on that as well, but I'd be happy to share any or all of it with anyone who's interested. Thank you.
Okay, thank you very much. I think we have time for 1 or 2 questions. Okay, well, thank you very much. So we now have time for a break before our final keynote speaker of the day. There are snacks that have been put out in addition to the lunch. Now there are now wonderful snacks, drinks, so please help yourself. And we'll start our last keynote at 3:15, so that'll be in about 15 minutes. So you've got a break for a little bit of time.
It gives me great pleasure to introduce Dr. Deborah Duran. Dr. Duran is the director of the National Institute of Minority Health and Health Disparities, Office of Science, Policy, Strategic Planning, Analysis, Reporting, and Data. She has 20 years of experience in organizational science policy. She has over 20 years of experience in organizational strategic planning, system assessments, science policy, measures, metrics, data management, performance monitoring, and reporting. Dr. Duran leads 2 offices within the office, including science planning, policy and reporting, and data assessments, resources, and evaluation. Here she serves the planning, assessment, analysis, and reporting needs and coordinates trans-NIH minority health and health disparities planning and reporting requirements. Dr. Duran hopes to help the NIMHD, or the National Institute for Minority Health and Health Disparities, become the centralized source of minority health and health disparities biomedical data, politics, and scientific advances. She has spent much of her NIH career serving as performance director in the NIH Office of the Director, handling a wide range of responsibilities including program performance monitoring, budget performance integration, organization performance assessments, and strategic planning. She designed a centralized online reporting system currently used by the NIH and the U.S. Department of Health and Human Services to assist in the collection, analysis, communication, and organization performance information. Dr. Duran trained in social psychology and virus research, statistics, evaluation, counseling, and computer science. She has experience as an educator, principal investigator, advocate, researcher, consultant, and counselor. Her areas of interest include science, system science, population health, Hispanic health, behavior research, cancer, coping, end-of-life care, palliative care, data systems, and training minority youth. Dr. Duran has a PhD in social psychology with a minor in research methodologies and statistics. In 2000 and also in 2004, Dr. Duran earned the Health and Human Services Secretary's Award for Distinguished Service. She is here today to discuss with her big data for or against health disparities. So let's give a hand to Dr. Deborah Duran.
I know this is the last session, so I'm going to make this as quick as possible. One of the things that I want to talk about today, though, is the importance of having the right data and being able to look at the data in the right way. And what I mean by that is having an objective eye. So, for those of you who don't know, our institute, you saw one this morning. NLM is one of the institutes at NIH. NIH is made up of 27 institutes. We are one of them, and our focus is on population health, on minority health and health disparities. So one of the things that we try to do at our institute is look at how does science impact racial ethnic groups, which are the top OMB defined. The bottom ones with the little stars is we also are responsible for health disparity groups. And the health disparity groups are populations that actually have a difference in their health outcomes that are mitigated. And these populations that are defined for us are low SES, rural, sexual gender minorities, and we also take in others that are of interest, like disabled and veterans and prisoners. I mean, there's quite a few. So one of the things that we've done is really try to clarify what is the difference between minority health and health disparities, and that's important when I get to talk about the data. The most important part that we did in the recent years was actually measurement-wise come up with what are the outcomes that define a disparity in health, which has never been done before. So this was just actually released in the American Journal of Public Health last month. And it's the first time that we've actually had this kind of metric to look at what is a health disparity, which then helps us when you start looking at data. In looking at this dynamic that we look at is, you can see the top corner, you have the health effects, but the colored section right here, these are all the types of things that mitigate what happens in a health disparity. So these are data points that help us to understand why the health outcomes end up being different for the different population groups. And as you take a glance at them, I'm going to pause for a moment because you're going to see there are many of these things that we never even measure. And if you're into data, if you don't measure it, does it exist? It doesn't. Yet we know it does. So this is part of the challenge that we have. And a lot of these are what we call the social determinants of health. And these social determinants of health often are the things that impact our health beyond biology. We know there are biological differences between the racial ethnic groups. We have different gene variances, we have different metabolic digestive systems, it goes on. And as science is unveiling these, we see these more. However, nothing that happens in your biology happens by itself. So it's all mitigated by the social determinants and what's in your environment. So we know that education is the primary cause of health disparities. It's hard to tease out poverty from education because they're so correlated. The less education you have, you tend to have more poverty. And so those two things are very important. But there's many things in our environment that also impact health disparities. So the reason we're interested in this is because, one, we want to make sure that in all of the big data sets, like all of us, like you heard this morning, and all the other data sets that you've heard today, that there is a fair and representative sampling of the population included. That's not always the case. The other reason that we are invested in this is because we also want to make sure that these social determinants of health are also included, because if you don't measure it, it doesn't exist. And we have to see what this is. So for instance, if a Black woman living in rural Mississippi is gay and she's in poverty, look at all of the variables that exist in her health and that intersectionality. We have to understand what the impact of her health is going to be for all the different isms that she now has. because we know that every ism that is there compounds her poor health, the probability of her poor health. And this is the other reason we care about this, is so that we have better clinical outcomes, so that we understand in the delivery of services which ways we should be treated. So, why do we need to pay attention to this? Because the demographics in the United States are changing. Right now, we are in California and New York, already the children entering kindergarten are 50% minority. Right now, approximately 60% of the population is white. That means 40% are other minorities. So if we're only basing clinical care, clinical trials on white people, where is that going to leave us with medicine? African Americans are about 13%. Hispanics have risen to 18%. Asians are around 13%, are 5.8%. Alaska Natives are 1.3%. And what we can count of the LGBT community is about 5% or 9 million Americans. In poverty, 13.5%. And rural people are about 20% of our population. So these are pretty substantial millions that we have to pay attention to. Hispanics are the largest population group today of minority groups. As you can see, the Asian, Hispanic, Black, and white. Blacks are actually pretty stable, but they look like they're declining because Hispanics and Asians are increasing. However, Hispanic immigration is at a decline. It's not because of the wall. You can tell by the dates. Okay? Part of that is also because as different countries have their own economic availability to make a living there, they stay. I got sidetracked here. The population is becoming more diverse, and as you can see in the projection by 2065, whites are going to be the minority. So it's important to have the data, just like the title of the presentation was, are you for or against? Because if you only have data on white people, pretty soon they're going to be the minority group. We also have an increasing, I don't have to tell you this because you feel it, right? The lower class is increasing and the middle class is shrinking. So remember, education and poverty are the two most critical factors of health. Guess where we're heading? Again, education has been pretty stable about the groups and their graduation rates, so this is not a major change of any sort. But the thing that is important, and this is, now I'm your mama talking to you, notice the difference in the income if you graduate. So all of you need to graduate because your income level changes. We pretty much know this from all the reports. Aging population, we're getting older and less children are being born. We've gone from the average child was 2.8, now it's 1.2 or something like that. Don't you ever wonder how that fraction child looks? I've always wondered that. What does a 0.8 child look like? I don't know. But it's important because we're getting old. And yes, I am. We're getting older and we don't have the young people to fill up the health gap. And so as you get older, there are more and more health issues. So it's more and more important that the data that we have reflect the population that exists. So big data. There's basically two types, structured and unstructured. In structured data, these are the types of data that we know a lot dealing like with the census. That's all the federal surveys where they have defined sets of questions that have gone over and over the different ways to ask those questions. And in those surveys, they tend to collect both race and ethnicity as well as some of the social demographics. So the structured data has sort of led the way into collection of data. Unstructured data is more new, although it's been around for a while, because structured data is like, or unstructured data is like Alexa, right? Artificial intelligence. I don't know if you guys, but my Alexa sometimes is so dumb, I can't even get her to tell me certain things because it's still a new field developing, but it's critical in where we are going, both in medicine, in criminal, like some of the data sets they were talking about. All of that is becoming AI. You're not even going to be able to walk through an airport anymore and have to do everything because you're just going to be scanned. That's why these things are so important. So this is an example of structured and unstructured data and how the unstructured data has sort of emerged from the structured data. In other words, the structured data existed first, and then people wanted to make it easier, simple, and faster, right? So they made the computers do it and not the people. I'm for that. So if you look at the structured data, though, look at the history. It started back really in the beginning of 1960s. That's when Americans reported their own race. But in 1980, and back in those days, it was only black and white. I think at one point it was actually Negro and white. And then as time evolved, the other races got in, but notice not until 2000. Scary, huh? Well, you guys were probably just born then, but it's not been collected it's only been collected for a short amount of time. And if you think of the importance of data, that time frame is not substantial. It wasn't until the last census that we even had mixed race, which there are many of us, if you've done your ancestry and all of us, we're all pretty mixed. Yet it's never been collected. The reason that's important is because you can look one way, which is what we call your phenotype, right? You can look, well, I'll use myself for an example. I'm Mexican. I can pass as white. Because when people look at me, they're going to assign me as white. But inside, and my DNA shows it, I'm Mexican. And Native American more than I am Caucasian. But I will, because of the way I look, I'm going to be classified as white. So then when they go to do my health care, it's going to be very different for me because they're going to treat me like I'm white, and I'm not white, right? So phenotype and genotype, these types of things are very important as we go through this in the collection of this data. It is a gold standard in structured data to self-report. In other words, when they ask me, I say, I'm Mexican. As opposed to me walking up to a counter or something and someone says, checks the box for me, she's white, because I know that's what they'll do. She's white. So the self-report became the gold standard. In structured data, it's not the gold standard in artificial intelligence and unstructured data. The ancestry biomarkers are like what you get in all of us. That's the genotype and stuff that's inside of you. And sometimes your phenotype does not match what your genotype is. So in unstructured data, you heard all of us this morning and the genome projects and stuff that's going on. 2-3 weeks ago, this hit science like a bombshell. I think probably most of you know that, that Henrietta was the first African American that gave her DNA. And so then she was classified. All the cell lines for African Americans came from her. Less than a month ago, they found out that her DNA had a whole lot of white in it. And so they misclassified her DNA and just called it African American instead of understanding what that cell line was. That's an important part in medicine, because keep in mind what all of us is trying to do, what you heard this morning. The intention of all of us is not just to collect genes, right? That's not what it's about. It's being able to diagnose from a cluster, from a genetic variation, what is a disease and disorder you have, and then what medicine will treat that disease and disorder. So if you're collecting cell lines and calling them African American, and they're not, I know you guys may not have graduated yet, but I think you know the answer to what that's going to do, right? The medicine is going to be off. So a perfect example of this is African American women who get breast cancer typically get a breast cancer called triple negative. White women tend to get a breast cancer that's HER2 positive. All of the treatment, let me back up. When breast cancer was a major issue for all women, the #1 cause of death, both black women and white women trajected up like this. And then there was all the Susan Love and the movement of breast cancer, got tons of funding, and they created a cure for HER2. So they can treat white women's breast cancer, and pretty much survival's really high these days for breast cancer. And guess what the black woman's gonna do? She's gonna die because the breast cancer treatments are more for the white type, not the black type. So then all of a sudden a health disparity happened, because now the white woman is getting treated because now they have specific to her type, and the black woman continues to project. This is why it is so critical that these systems collect the right data for the right people. The last line, every time that there's a misclassified cell line, it is inadequate to understand the disease of that population group, and therefore then the treatments to treat that disease. And as that occurs, more and more health disparities will exist. This is an example. I don't know if she showed these to you this morning, but this is the most recent data of what is actually in all of us and the genome collection. And if you notice that Europeans in in 2009 was 96%. In 2016, it's 81%. Do you remember the slides in the beginning of how many minorities there are in the country? There's kind of a mismatch there, right? And look at it, and of the minority groups, Asians are the highest. But do you remember the statistics on the beginning up there? They were among the lowest in the population in the United States. What do you think that is a result of? Sampling, right? Convenient sampling, because they're in the sciences. So if we're not being represented in these types of data sets, medicine's not going to help us. What we found in the data sets is that the other sort of biases are that they collected off of people that excuse me, that tend to be sick. Once again, convenient sampling, right? Because they're right there. It's also the demographics around the hospital, because who works around the hospital? People that work there. And so they're invested in it. So again, convenient sampling. Clinical trial enrollment. So we fight this battle all the time. For any of you who also do any kind of medical records data and clinical trials, minorities are excluded, or they're not included in clinical trials. Down here you see that 2% of 10,000 NCI studies contain adequate sample of racial and ethnic groups. So we're setting up these databases not to reflect the United States. So in artificial intelligence, it's booming. It's like it's taking off because it actually works so effectively. It can predict. So we're using it in medicine in what they call chatbots, oncology, pathology, and rare diseases, because we're finding that it can actually predict even better than physicians can of what disease, particular, like cancers, or they use it for many things. But the reason I want you to understand how fast it's growing is because of the next slides. It is predicting disease and disorders, but it's leaving out a whole lot of other people. So one way, so this is not health. This is another form of the artificial intelligence that they're using to map release, you know, like when they release prisoners, and then they predict whether that prisoner is going to get put back into jail, and then the neighborhoods they will reside in, and then what will happen with the crime in that neighborhood. And in this database, so we use it in medicine, we're using it in crime, it over-predicts what African Americans are going to do and under-predicts what whites will do. Skin cancer. African Americans have the highest mortality for skin cancer. Not necessarily the highest rates, but the highest mortality. White people have the highest rates. But skin cancer is pretty easy to cure if it's caught in time and if it's dealt with, okay? So they're using artificial intelligence for this, but in the artificial intelligence that they use for women of color, it can only predict it 34% of the time. For white males, it was 99%. So the darker the skin, the less it can detect skin cancer. Another big system, this actually got put on hold, the weve. It's like what they were going to use it to like get through the airports, right? And so they were scanning to see as a clearance. Again, a person's gender identity, 99% white men, they could do. 35% dark-skinned women, they couldn't. That was the only prediction rate. When they did a survey of Google, looking at like kind of like a LinkedIn thing where they search through to find people who can apply for a job, it favored men over women for higher paying jobs. So it's not just about race, it's also about gender. If you notice the other algorithms up here, when they set some of the algorithms in AI, they've mistaken images of blacks for gorillas. They've misunderstood Asians to be blinking when they weren't. They judged only white people to be attractive. The link showed a preference of male name searches, especially for high-paying jobs. And they learned from Twitter and others kind of spouting that the perpetuation of anti-Semitic messages. So AI is convenient, it's rising, and there are some issues. And part of the problem with this is because of the second bullet. What's getting programmed into AI are white, urban, middle-class males. So we call this the weapons of math destruction. And it's just like in any other data set, right? Garbage in, garbage out. That's pretty simple. It's not the computer's fault that it's only predicting white men 99% of the time. It's not the computer's fault that it only can identify a black woman 35% of the time. It's not the computer's fault, right? Because garbage in, garbage out. Good data in, good data out. What are we doing? We're making something that's supposed to be objective and fast, as biased as we are as human beings, because of the way that it's getting programmed, right? In artificial intelligence, they have to program it over and over and over again. Well, if you keep putting the same stuff in as your own bias, it's going to give you the same bias in a computer that is supposed to be objective. So if you take nothing home from me tonight, or today, still light, use data with caution. Look at how it got created. Look at where the variables, what variables are assigned to it, so that when something gets spewed out, you know where the biases are. I'm not saying that data is bad. I use data every day of my life. Data is strong, it's got patterns, I love data. But you have to know what its limitations are and not perpetuate the damage that a data system that has biases will produce. One of the things in the artificial intelligence, they program it with patterns. And in these patterns, we can change these patterns by making sure that there's a diverse audience that also gets put in, right? So let's take the dark skin cancer piece. You need more African Americans with skin cancer to get what it looks like to feed it into the system. And then the system will predict for the African American with skin cancer just as well as it can for the male. But you have to be conscious enough to know how to do that, right? Or that you have to do that. Oh, by the way, FDA has just recently, the bottom bullet, is realizing that this is a problem. So they are now requiring and paying close attention, just like I just said to you. Pay attention to your data. They're now paying attention to systems that don't do this adequately, which is a good thing. Government is good. So the most important part down here is the last bullet, is that if all of the racial and ethnic groups are included into these data sets, then the disparities are going to get reduced, right? If they're not, the disparities are going to continue to rise. And you know what? I'm predicting they're even going to go double and triple in the next few years unless this data gets changed. Because more and more, NIH is developing, we fund many people, the University of Cincinnati, to develop medicines according to the genome, according to your biology, according to your personalized medicine, right? But if you have no more minorities, you don't understand the effects of poverty or education, guess who's going to get cured? Guess who's going to die? And it's going to get worse and worse because that is where medicine is going. So one of the things in the first bullet is what I just said, that this is such a serious problem that it's not just a matter of, oh, we should just collect it because it would be a nice thing to do. It is a matter of life and death. And when we don't do it, we're just perpetuating the biases that we already have as humans. And we can treat, it is not ethical to treat one population group better than another population group in medicine. Here's another example. It's not just about the data. So I'm asking you also to pay attention to the policy of how that data is used. So in kidney transplant, and for those of you who don't know, African Americans have the highest kidney failure rates. They have a gene, APO1, that actually contributes to that. It's very common. We know this is a very common condition. They end up needing kidney transplants oftentimes in their 30s and 40s. So very young, their kidneys start to fail because of this gene. This is the exact policy of what the kidney transplant requires. So you have to be young. If you're a child, you're higher on the list, right? You know, in transplants, there's criteria that have to be met. So if you're young, you're higher on the list. A person who gave a kidney away also gets priority on the list. And so then they also have to have a certain number of matches, but also there's not a lot of diversity in those kidney matches either, in donors, so that's also a problem. And the machine, so this is not done by hand anymore. This is done by artificial intelligence. This is done by a calculation, okay? So what this means, by the calculation of this policy, that a 65-year-old white woman is expected to live 20 years longer are to live another 20.5 years in 2015, 4 years longer than a black man of the same age. So by the algorithm that policy is going to do, who's always going to get the kidney? The white woman. Who needs the kidney the most because they have the biggest population problem with it? Black men. So you also have to pay attention to the policies of the data sets. Now, how do we go about making this better for us? We have structured and unstructured data, right? We know that in structured data, we've got it put together. So with race and ethnicity and social determinants. So let's combine the two. Let's fuse them together. Put together structured and unstructured. Here's an example of a study that actually did this. So it was a Google search, and they looked for, media twitting and all these things, they looked for where the N-word was used. And they saw the incidence of that, and then they matched it with the structured data, and they found that in the neighborhoods in which blacks resided the most, the N-word was used the most, therefore racism prevailed in those areas. When you look at the data of those areas, they also have the areas of the poorest health for blacks. This is another study where they combined the two. They did environmental, and they looked at where the minorities lived, and they concluded that what is happening is minorities are being pushed into or living in high pollution neighborhoods where a lot of industry and toxins are in the air. So again, a #1 cause of health disparities and poor health. So the social determinants are an important part of the data system that we need to combine with the other things, whether it's all of us, artificial intelligence of any sort, because these things help clinicians also understand how to treat their patients. If you, for instance, I use the example of breast cancer, if the doctor doesn't realize, they do, by the way, if they didn't realize that that treatment is only for white women, and they give it in the same doses in the same way for black women, all it does for her is makes it toxic, and she's worse off. So they have to adjust the treatment because they understand that there's a difference. If a clinician wants a person to come back and get a treatment every week, yet they don't have the money to get back to the doctor's office or the money to do the co-pays for the treatment, they're not going to likely do it because they can't afford it. So then they have to adjust what they do in the care of that patient. So these social determinants are also just as important as the person himself or herself. This is a couple examples of that combination of poverty and biology. And every time we see this combination, we see the impact of it on health. So the social determinants are also just as important. Why do we care about this also? Is because we look at the labor force. They don't have statistics really about where the data scientists are, or like how many we have in the United States, but we have student data. And as you see the student data, the minorities aren't going into data science. And if the minorities are not going into data science, then they're not raising the questions to be sure that the racial and ethnic groups are there. So those of you who are not minorities, now you know to raise the question. We do many different things at NIH. This is a data system that I just actually launched this year. I would suggest you guys go on it anytime, add this to your repertoire of data. It has racial and ethnic data, and it has social determinants of health in it. And it's done at a county level. It's called HD Pulse, and it shows health disparities and different demographics throughout the United States, only the United States. Some papers that we've done, we're working with Phoenix, which is another genomic kind of database, to be sure that a core set of data elements are collected. And then if this is online or published anywhere, these are grants that you can apply to deal with data science and do studies and make the systems better. And there's 3 pages of them, okay? Questions?
Yeah, thank you. So my name is Dominique Brandt. My question is that I heard you saying the government is good, is efficient. So you talked about research, data analysis. You talked about the government, but there is another group, which is the pharmaceutical industry. So how would the government help the pharmaceutical industry provide drugs for minority, which are still minority now, meaning the number of, they are not going to make their profit out of drugs for instance, your cancer drug for skin cancer, for the number is not big, because I think one part of the problem is the number, is that for now, white people were able to afford to pay for it, and this is why research was targeted to this area. So is there a reflection of supporting the industry towards this type of drug?
So we do lots of government does lots of partnerships with pharmaceutical companies. You are correct that they do have a profit margin in mind. There are some pharmaceutical companies that have morality, so they also invest some in what we call rare diseases, which does not have a big profit margin for them, but they have morality, some of them. NIH funds many things with them, but again, you're correct, their job is profit. And that's why it's also important, like NIH, to exist, because our intramural program, we don't care about profits. So we do things that are rare disease. We do things that pharmaceuticals won't do. We do things so that there's health for all of Americans. And we do work with them and try to make sure that they have the right variables in paying attention to it. And they're learning the hard way anyway. Right? Number one drug that I can tell you about with my point is warfarin, right? That it doesn't work on African Americans. So if they want to make a profit, they also have to develop drugs that works on the other 50% of the country, right? Because their profit area is narrowing. So as whites become 40%, they're going to start paying attention, right? Did that answer your question? Okay.
Just on that point, may I ask then, because you're thinking of percentages of Americans, but of course, if we look at the number of Latinx or African Americans who carry genes that may actually represent a larger part of the percentage of populations outside the United States that could benefit. if you want to think about profit margins, it can completely change if you think about the rest of the world. So how much of what you're talking about is actually looking at populations beyond just, I mean, the percentages that exist for the United States may not represent actually the world population?
Well, I can tell you that NIH invests in international research as well. We have a whole institute that Fogarty does international. NIGMS, which is genomic, does lots of studies where they look at the gene variances within the United States and the mother country of wherever someone is. So we invest money and time in those things. But you don't, and there's sometimes that we have to go to those countries to do something. So you guys know Gardasil, right? Where you're supposed to give it to your adolescent children now, the cervical cancer. There wasn't enough cervical cancer in the United States to develop that. There enough N. So it was actually developed in Hispanic countries because it's more prevalent there, so they could develop it there. They could use the analyses there. So it's not like we're blind to the whole world, but we focus a lot on what is done in the United States because we kind of have all the gene pool here. All the races are here, all the biologies are here. And when it's not, we go there. I can't, we don't do pharmaceuticals. Any other questions? And every time you look at a data set now, you're going to question it, right? Shake your head. Good. Mission accomplished. All right, thank you.
Thank you. I'll just go. Okay. All right, we have come to the end of our day. I do want to give another thank you to Dr. Duran. When she and I first spoke on the phone, we talked about just simply the importance of including minorities in research studies and just how important it is for researchers who are looking at the data and the outcomes of those studies to understand how that simple inclusion might impact the data. But she's taken it to a whole nother level and talked about simply when we look at artificial intelligence and how that data can be skewed just simply based on who took the research and who's responsible for collecting the data. And so I want to thank her for that because I think she's opened a lot of our eyes to some of the things that we probably took for granted and some of the things even myself as a minority, sometimes I'm thinking as accurate or valid that may have not even included anyone that looks like me. So thank you for bringing that to light. I appreciate that. So I want to give you another round of applause. I want to thank our audience, those of you who came at the beginning of the day and who stuck and stayed, those who came at some point in the middle of the day and who are still here with us. We appreciate you because obviously we could not have had a successful event without our attendees, so thank you for coming. Thank you for partaking and asking questions. The whole idea of this particular theme that we've selected was one to attract. It was a provocative theme on purpose, and we wanted people to start to think about how these issues play a part in the information that we receive and that we collect and that we take at face value, and how it impacts how we go about our daily lives. And so hopefully the themes that we've done today, the talks and the discussions that we've had, whether it was about health disparities, things that we may not have considered when it comes to gender and how people identify with certain genders, and how the Cincinnati region and how Ohio is dealing with health disparities, on down to how minorities and certain ethnicities are impacted in our justice system. And so it's been a very full day. So again, thank you for staying. I want to thank, in particular, two people, Lindsay Nichols and Stacy Denson, who have provided our accessibility resources. So they are the two ladies that you've seen up here. They've been doing a phenomenal job behind the scenes, and if you haven't seen them, we've definitely been able to kind of see them in action, and it's a tremendous thing that they're doing to be able to help those who are impacted by what we've been talking about, actually kind of take part in the ideas and things that have been exchanged today. So we definitely appreciate having them today. I would ask that all of the members who are still remaining of the Planning Committee stand. Get up. So I am going to call them by name, and if you would just wave your hand, we just want to acknowledge you. I appreciate all the work that everyone's put forward to make this day successful. So just starting from the front, we have Jane Combs, Amy Latessa, we have Richard Johansson, Mark Chalmers. These two gentlemen were also responsible for the power session, so those of you that attended, let's give them a very special round of applause. At our library table, we have Amy Koschoffer, who is my partner in crime, and Lori Harris. Missing because there were some other duties that they had to attend to are Ted Baldwin, Don Jason, and also Rebecca Olson. So they are not here, but they were very much present during the overwhelming majority of the day. So we appreciate you all for your work that you've done as well. We want to give one final shout out to the National Network of Libraries of Medicine, Greater Midwest Region, for their funding and their sponsorship of today's programming. Also for UC's very own Office of Research for their sponsorship as well. We appreciate them, so one more round of applause. And I guess last but not least, all of the videos and all of the presentations have been recorded. So for those of you who would like to go back and review the information that's been presented, you will have that ability to be able to do that. We've also live streamed, so thank you for those who are still sticking and staying with us. We appreciate you for taking the time out of your busy schedule. Definitely also want to thank the keynote speakers, those who traveled, especially the keynote addressees, as well as our panelists who traveled as well. So some of them actually came from Columbus just this morning. So we appreciate that as well. So with that, we will say that it's a wrap for our 4th annual Data Day. Thank you.